Welcome back to your second lecture, Chapter 2, which covers both the chemical and physical properties of water and also serves as a review for acid-base chemistry and buffers. In class, we will then apply the content covered in the lecture video and the textbook to Activity 1 from your POGIL workbook. This activity is going to allow us an opportunity to explore and discuss factors that govern the characteristics of biomolecules in aqueous solutions, as well as give us opportunities to practice applying problem-solving methodology to make a credible biochemical argument and to further develop skills associated with teamwork needed to produce a high-quality response to a challenging problem. Here are the learning objectives for this chapter. Please be sure to pause and read over these objectives before proceeding with the video so that you have them in the back of your mind as we work through the material. Again, keep in mind that you can find these PowerPoint slides on the Moodle course page. Water is very important, especially in biochemical reactions, because one, almost all biological molecules assume their shape in response to water. Remember, we talked about this a little bit the other day in class when we were talking about the hydrophobic effect. The medium for the majority of biochemical reactions is aqueous-based, and water participates in many biochemical reactions either as a reactant or as a product. A water molecule consists of two hydrogen atoms bound to an oxygen atom with a hydrogen-oxygen bond distance of 0.95 angstroms, with a bond angle of 104.5 degrees. Now, this bond angle is actually really interesting because if we take into consideration the lone pair on oxygen, we could say that water has a tetrahedral geometry. Now, typical tetrahedral molecules should have a bond angle of 109.5 degrees. However, this bond angle of water is smaller because the lone pairs that take up a lot of space on oxygen, pushing the two hydrogen atoms closer together. Because of this phenomenon, we actually end up with a stronger dipole moment in which the oxygen atom has a partial negative charge and the hydrogen atom has a partial positive charge. The presence of the dipole moment created from the perturbed tetrahedral geometry of water is what allows water to make relatively strong hydrogen bonds. A hydrogen bond is where a hydrogen atom covalently bound to an electronegative atom such as oxygen, nitrogen, fluorine, or sulfur interacts or is drawn towards another electronegative atom. In addition to the hydrogen bonds forming in water, we see hydrogen bond formation in a variety of biomolecules such as proteins and nucleic acids. And often these interactions are one of the many stabilizing forces behind secondary and tertiary structures, which we'll discuss in more depth in Chapter 4. When we look at the hydrogen bonds that form between two water molecules, the average bond distance between the hydrogen bond donor, or the hydrogen, and the hydrogen bond acceptor, oxygen, is 1.8 angstroms and requires approximately 20 kilojoules per mole of energy to break apart. When compared to the 460 kilojoules per mole of energy required to break the covalent bond between oxygen and hydrogen, a single hydrogen bond may not seem like a large amount of energy. However, when you have multiple hydrogen bonds within a structure, as seen in an ice cube, it can add up. When we look at ice compared to liquid water, we see that the water molecules orient themselves in such a way as to optimize the hydrogen bonds. Each molecule of water in ice is able to hydrogen bond with 4 other molecules of water, creating a crystal lattice of water molecules. This rigid and highly organized structure derived from the optimal hydrogen bonding is what gives ice a lower density than liquid water and allows ice to float. As water melts from ice to liquid water, we begin to see that the hydrogen bonds become much more dynamic, allowing the molecules to still form hydrogen bonds, but they fluctuate from molecule to molecule as each molecule passes each other by. Although these hydrogen bonds are transient, they still provide some stability to liquid water, which can be observed on the relatively high boiling point of water, which is 100 degrees Celsius, compared to other liquids with similar molecular weights, such as methane. In addition to hydrogen bonds, there are a number of other non-covalent interactions that are very important in biomolecules. These interactions include ionic interactions, dipole interactions, van der Waals interactions, and the hydrophobic effect, which again, remember we discussed in class. Examples of these various non-covalent interactions can be seen here in Table 2.5 of your textbook. So do feel free to review this if you need a review from organic chemistry and the non-covalent interactions that functional groups can form. Anytime we talk about water and biomolecules, we need to define and compare the terms hydrophilic and hydrophobic. Hydrophilic molecules are polar and/or ionic materials that can easily dissolve in water and is stabilized further through hydrogen bonding and dipole-dipole interactions. When we dissolve salts in water, we actually create a cage-like structure of water molecules that create a solvation sphere and orient themselves in such a way as to have optimal hydrogen and ionic interactions. Hydrophobic molecules, or nonpolar water-fearing molecules, do not like to dissolve in water and tend to aggregate in an aqueous environment in order to minimize the surface area exposed to water for each individual nonpolar molecule. By aggregating multiple nonpolar molecules, we can release what we call trapped water molecules back into the bulk water, increasing the overall entropy of the system. This release of trapped waters by aggregation or the folding in of hydrophobic residues is what we call the hydrophobic effect. The hydrophobic effect can also explain why we see amphiphilic molecules, or molecules that contain both hydrophobic and hydrophilic segments, to fold or assemble the way they do in an aqueous environment. A classic example in biochemistry is the spontaneous micelle formation of fatty acids in water. These molecules, when exposed to water, will spontaneously aggregate together to form spheres where the hydrophobic alkane chains are in the center and the hydrophilic carboxylic head groups are on the surface. In the core, we have favorable hydrophobic van der Waals interactions between the tails, and on the surface, we have favorable hydrogen bonding between the polar head groups and water molecules. In addition to the hydrophobic effect during the formation of micelles from single-tailed fatty acids or phospholipids, we see the same effect in the assembly of the lipid bilayer with two-tailed lipids, such as the plasma membrane of cells. Instead of forming a circular sphere, we form a bilayer in which the core is filled with the carbon-hydrogen tails of the lipids, and the inner and outer surface of the membrane is comprised of the phosphate head group. We will address why we get a bilayer versus a micelle in a later class activity, but both structures are driven by the desire to bury the hydrophobic regions and expose the hydrophilic regions when in an aqueous environment. When we begin discussing water and cellular membranes, we also need to discuss and differentiate between the terms osmosis and diffusion. In osmosis, we have a membrane that is permeable only to water. If the inside of the membrane contains a higher concentration of some solute, such as a salt, water will then move inside the membrane in an attempt to equalize the concentration of that solute on both sides of the membrane. However, if there is no solute present outside of the membrane, we will never reach the same concentration on both sides, regardless of how much water moves into the membrane. Eventually what happens is that so much water moves into the membrane until there is so much pressure that we reach a movement equilibrium of water in and out of the membrane. And this pressure is what we call the osmotic pressure. Diffusion, on the other hand, is when solutes can freely cross the membrane. For instance, in the common biochemical technique of dialysis, a membrane is used with a specific pore size in which molecules that are smaller than the pores can freely pass between the external and internal environments, while larger molecules are still trapped inside. This technique allows biochemists to exchange salts and buffer conditions without diluting the biomolecule they are trying to study. Cellular membranes are semi-permeable only to water. Cells that are isotonic have an equal flow of water in and out of the cell because the concentration of salts inside and outside of the cell are near equal. Should the environment outside the cell decrease significantly in salt concentration, water will begin to move into the cell faster than it moves out, causing the cell to swell and burst, which we call hypotonic. If the opposite happens, in which the concentration of salt outside of the cell increases, maybe due to dehydration or high dietary intake of salt, more water moves outside of the cell than inside and the cell shrivels up, and we call this hypertonic. Another interesting characteristic of water is its ability to act as a weak acid with a pH of 7. It is able to both donate and accept acidic protons. We can also compare the pH of water with some other common liquids, such as acidic gastric juices or basic ammonia. But if you look around the neutral range, you will see milk, saliva, blood, and tears. Indeed, the ideal pH range for biological fluids is around pH 7. This pH is the optimal pH for enzyme activity and the proper folding of biomolecules and must be strictly controlled. However, in the cell, we have a myriad of chemical reactions that can produce both hydronium ions and hydroxides that can affect the local pH. Now, like I said, water itself is a weak acid, but it's not a very good buffer. In order to maintain the biologically optimum pH of 7, the body utilizes buffers. Buffers are mixtures of weak acids and their conjugate bases that can resist fluctuations in the pH. When we have an equal mixture of the conjugate acid and the conjugate base, the pH is equal to the pKa, and we have the greatest buffering capacity. Buffers work because as small amounts of acid or base are added to the system, the conjugate acid-base pair can consume the added hydronium or hydroxide in order to bring the pH back to equilibrium. However, the buffering capacity is lost if we consume one of the two pairs by adding too much acid or base. Typically, this happens if we go 1 pH unit above or below the pKa. One example of a buffer is the weak acid acetic acid. In this system, as acidic protons are added to the solution, acetate is converted to acetic acid. But as we move back to equilibrium, the acidic proton is then donated to hydroxyl groups that can also be added to the system in order to make water, neutralizing the solution back to the pKa of the buffer. As you can see in the titration curve, the buffering range of acetic acid is around its pKa of 4.76, which despite increased concentration of hydroxide to the solution, it stays relatively constant until you break the buffering capacity around pH 5.76. Each weak acid can have a different pKa, such as we see with the phosphate and ammonia. And the buffer that a biochemist selects depends on the pH at which you want to maintain. But the most common buffer that biochemists use is the phosphate buffer, which has 1 pKa around 6.85. I say phosphate buffers have 1 pKa around 7 because phosphate buffer is actually a polyprotic acid in which it has 3 donatable protons and therefore has 3 pKa's. In this instance, you would have to make sure you are using the correct conjugate acid-base pair around the pKa with which you are targeting. Now, biochemists can fine-tune the pH at which they want to operate by mixing different ratios of conjugate acid and conjugate base. To do this, we use the Henderson-Hasselbalch equation in which the pH is equal to the pKa plus the log of the ratio of the base over the acid. This equation is also useful if you're trying to determine the percent protonated species of an acid-base pair at a certain pH. This gets to be especially important when we're talking about enzyme and enzyme mechanisms, and we'll get some practice doing this next week. One important example of a biological buffer is the bicarbonate buffer found in the blood. In order to maintain both the pH of the blood between 7.35 and 7.45, as well as facilitate carbon dioxide transportation out of the body, the blood uses the bicarbonate buffer. We will visit this again in more detail when we talk about hemoglobin, but I just wanted to bring this up as an example of how a buffer can both be used to maintain a certain pH in the body, as well as do a secondary job, such as transporting metabolites out of the system. All right, so again, make sure you guys look over your learning objectives and do your muddiest point questions on the discussion board on Moodle, and I will see you in class. Bye.